Morning, everyone. Thank you for attending this seminar given by Professor Kimberly Kline this morning. So this seminar is jointly organized by the School of Biological Science and Institute of Advanced Studies of NTU. Now this is actually a pre-symposium talk. You know, for many of you, you probably know we were trying to organize this symposium called Frontiers in Peptide Science and Drug Discovery in honor of Prof. James Tan. But you know, this symposium was supposed to take place in February, but because of COVID-19, we had to postpone it. Actually, everything was ready. You know, the wine bottles were delivered. I mean, in the end, we have to return them back. So the questions, after the seminar, the questions will be addressed. You can ask questions by pressing the raise hand function and at the same time unmute yourself, or you can leave your questions in the chat window. For now, let me give a brief introduction to our speaker, Prof. Kimberly Kline. For those of us from NTU, everybody knows her. Kimberly is an Associate Dean for Faculty Affairs in the College of Science of NTU. She is Associate Professor in Microbiology in the School of Biological Sciences and a PI at the Singapore Center for Environmental Life Science Engineering. So before coming to Singapore, Kimberly received a Master of Public Health in Biostatistics and Epidemiology and a PhD in Microbiology and Immunology from Northwestern University. Then she conducted her postdoc studies at Washington University in St. Louis and also at Karolinska Institute from Sweden. And Kimberly has received multiple awards for her work, including an NIH K99 Career Development Award in 2011, the Singapore National Research Foundation Fellowship, and the ICAAC Young Investigator Award from the American Society of Microbiology, and also Nanyang Education Award in 2017. So in addition, Kimberly is an active advocate for increasing diversity in science, technology, engineering, and mathematics. So in 2018, she co-founded this Women at NTU platform. So just last year, this initiative received a grant from the Ministry of Education here in Singapore for the promotion of women in engineering, research, and science at NTU. So today, Kimberly is going to give a talk on this topic, strategies to slow the development of daptomycin resistance in Enterococcus faecalis. So Kimberly.
Thank you, Chuanfa. And thank you all for showing up to listen today. So when I was invited to give this talk, Chuanfa asked me to talk about bacteriology and antimicrobial resistance. And so I actually had to do a little bit of work to tie all of those things together. And of course, because this pre-symposium seminar is partially in honor of James Tan, I needed to include something about antimicrobial peptides. So what I have done today is put together a new talk that I have not given before, but it's kind of a comprehensive talk for at least one line of investigation that's been going on in my lab since I moved to Singapore, and it's work that I actually started as a postdoc. And so what I think the way that we should think about this talk is a nonlinear talk, and this is denoted by this broken up arrow here. And it's more like a series of vignettes. And so what I want to do is start at the very beginning, sort of how we started thinking about this particular project, and then how we sort of progressed over time ultimately to end up at this place where we're now starting to think about antimicrobial resistance, discovering potentially new strategies for ways to slow antimicrobial resistance. And so that was the title of my talk. But the first half of the talk is not going to have to do with resistance, but it's more about basic microbiology, some fundamental questions that we've been investigating. And so I think one of the take-home points that I hope you leave this talk with is sort of the clear appreciation that really basic curiosity-driven science and research is really important for these downstream discoveries that might have practical and translational applications. And so with that in mind, I will get started here. So the bacterium that we study is Enterococcus faecalis, or the primary bacterium we study. And so for those of you that maybe don't think about bacteria as often as I do, this is a gram-positive bacteria. This is a bacteria that's of a big public health concern. And it's placed here on the Center for Disease Control list of serious threats in the United States. And it also shows up on the WHO's list of most concerning pathogens. And that's because these infections are often associated with bacterial strains that are resistant to last-line antibiotics, namely vancomycin, as you can see here. And so there's a lot of these cases, and they lead to significant death and morbidity as well. And so they're very difficult to treat. So there's two strains of Enterococcus that primarily infect humans. One is E. faecium, and so that's actually the one that's most frequently associated with vancomycin resistance. So up to something like 80% of E. faecium infections are vancomycin resistant. However, those infections are not nearly as common as the Enterococcus faecalis infections. So only about 10 or 15% of faecalis infections are vancomycin resistant, but those infections are far more frequent. So collectively, vancomycin resistance is actually important in both of these organisms. And so, thinking about then, what do we do when these infections or these strains are resistant to all the antibiotics and the last line antibiotics like vancomycin? So luckily we still have a few treatment options for these strains. And so these there's a handful of drugs that are used, but I think most frequently these are linezolid and daptomycin. And so I'm going to come back to daptomycin at the second half of my talk. So just hold this in mind. Okay. So Enterococcus faecalis and faecium, they're both commensals of the normal healthy gut microbiome in humans. So everybody that's listening to this talk probably has Enterococcus faecalis in their GI tract, and this is not a problem. This only becomes a problem in compromised or more susceptible individuals, people with underlying illnesses, comorbidities, generally hospitalized patients, where this bacterium can cause opportunistic infections. These are most frequently catheter-associated urinary tract infections and wound infections. And it's a variety of wound infections, surgical site infections, diabetic foot ulcers, and other types of wound infections. It's also associated with massive overgrowth in the GI tract in individuals that are undergoing broad-spectrum antimicrobial therapy. And it's also associated less frequently with infectious endocarditis. And so it's not one of the main causes of infectious endocarditis, but the mortality rate is actually higher than some of the other more frequent causes of bacterial infectious endocarditis. Okay, so why am I telling you all of this? Because to put this all in context, what we think about in my lab is how Enterococcus faecalis perceives all these different niches that it finds itself in the human body, how it responds accordingly to often change its metabolism or change, for instance, its surface structures to better cause these infections. And I want to just highlight these surface structures that are shown in the upper right picture here. These are pili, and I'm going to come back to these in a little bit. These are really important for adhering in the heart, in the urinary tract, and other places in the body. And so why do we think about all of these? So this is important for the context of this talk. We want to understand how the bacteria senses and responds to its environment to cause infection, because these are potentially interesting intervention points. If we can understand these processes, then we can try to figure out ways to disrupt them and to limit disease. So that's really our guiding principle in all of this research. Okay, so vignette number one. So for me, this all started with this really, interesting observation. And that's shown in these two pictures. So on the right, we have this picture of immunogold labeled scanning electron micrograph of Enterococcus faecalis labeled for this surface protein called aggregation substance. This is an old picture. It's always fascinated me. And this aggregation substance is this sticky surface protein. It's stuck to the outer cell wall of E. faecalis. And it's important for bacterial-bacterial interactions that are essential for horizontal gene transfer. So the bacteria have to stick together closely for conjugation to happen. And so aggregation substance is important for that. When we started studying pili, which are these long filamentous appendages you can see here with these gold labels on them, we noticed right away this really interesting phenomenon, which was that the pili were excluded from the septum of the cell, very similar to what we see with aggregation substance. And so this was really fascinating to us because both of these surface proteins or surface structures are attached to the cell wall, and they're done so by the same mechanism. And so we wanted to try to understand what underlies this interesting observation. So purely curiosity driven at this point. And so to address the question of why this is, we took a step back and wanted to look at the first step of how these proteins get attached to the cell surface. And so I need to give you a little bit of background about how that happens. So there's these enzymes in gram-positive bacteria called sortases. They are conserved in gram-positive bacteria. There's a slight variation version of these in some gram-negative bacteria as well. These are membrane-anchored enzymes, and you can see this sort of in this boomerang-shaped picture here. This is your sortase enzyme, and they act on very specific substrate proteins, like the aggregation substance I showed you and these pilin proteins. And in the very bottom, you can see what a typical sortase substrate looks like. It has an N-terminal signal sequence, and at the C-terminus, it has this cell wall sorting signal. And this consists of an LPXTG motif, a transmembrane domain, and a very positively charged cytoplasmic tail. And so what happens is that these substrates are targeted to the general secretion machinery, shown here on the left. They're translocated across the membrane, and then they're transiently held in place in the membrane via this transmembrane domain, and they're at the C-terminus. And so you can see this here on the left. It's what's called P2 in this figure. So that transient substrate is then recognized by the sortase enzyme itself. Sortase then can recognize that LPXTG motif. Specifically, it cleaves between the threonine and glycine, leading to this sortase substrate intermediate that's still stuck on the membrane. And then that transmembrane domain floats away. Subsequent to that, lipid 2, which is a precursor for the peptidoglycan cell wall, that's still in this model, still anchored to the cell membrane. There's this uncross-linked peptide cross-bit bridge that's part of lipid 2, and this amino group mediates this nucleophilic attack on this sortase substrate intermediate, leading to a covalent attachment of that substrate now to lipid 2. And then lipid 2 is transferred from the membrane to the cell wall and incorporated into the growing cell wall. And this is how these proteins end up on the cell wall. Okay, so you remember this was all driven by our interest in understanding how these proteins get in this really interesting pattern on the cell wall. So the first thing we wanted to do then is ask, well, are the proteins that are mediating this initial attachment, are they localized in the cell? And so to do that, we created some strains in which those sortase enzymes are labeled. And so what I didn't tell you is that Enterococcus faecalis has two sortase enzyme types. One is the type I just described to you that attaches the proteins to the cell wall. There's a second sortase, it's called Sortase-C, it's shown here in purple. That actually is involved in the covalent polymerization of the pili themselves prior to the anchoring of those pili to the cell wall by sortase-A. Regardless of which sortase we're looking at, when we labeled them and looked at where those proteins were localized, you can see that they're very discretely localized to domains at the cell membrane and often at the septum of the cell. And so this was our first indication that these processes are not randomly distributed, but are localized. And so we can see this also. So these first images were cross sections. So on transmission electron micrographs, we can also see this on whole cells. So here we're looking at sortase A, and you can see that as the cells go through the division cycle, we can see sortase A sort of at the middle of the cell pre-divisionally. And then as the cells divide into the late divisional stage, we can see these sortase enzymes moving to what will be the next septum of the cell. So these are often septally localized. So because sortases are acting on secreted proteins, we also looked at the location of SrtA, and which is one of the drivers of the general secretion machinery, and we see that SrtA localizes in a very similar pattern. And importantly, these enzymes co-localize together. And so this all started to then suggest that this forms a complex, a microdomain, if you will, in the membrane, leading to the assembly of these virulence factors. And so we did some work to try to understand what governs their localization of these proteins. And we looked at 3 different things: sequence motifs, molecular interactions, and anionic lipid domains. And so in terms of sequence domains, we started to think about this because when we looked at the sortase enzymes, and if you kind of zoom in at the cytoplasmic tails in these cartoons, you can see that these tails are very, very positively charged, much more positively charged than they need to be to dictate their topology of these enzymes in the membrane. And so here you can see this a little bit more closely. So here, SrtC, here's the cytoplasmic tail. So again, I think it's 13 amino acids in this cytoplasmic tail. 8 of them are positively charged. This again is more than that needs to be to dictate the membrane topology of the protein, and it's far more than would occur just randomly in a protein in the cell. So we wondered if these positively charged amino acids were important for the specific focal localization of the protein. We mutated them, asked what happens to this protein. And in fact, this does result in the delocalization of this sortase enzyme. And so we can quantify that here. And we see a really significant reduction in the number of cells that have single foci of this enzyme. So these positively charged amino acids are important for the localization of these proteins, but also it's important for the function, because in the strain where these sortase enzymes are mislocalized, remember this is the SrtC is involved in assembling pili. We also see a significant reduction in the number of cells that are pileate. So SrtC is localized, and when it's delocalized, it doesn't carry out its function as efficiently. So this is giving some reason why the cells might do this. Okay, so sequence motifs are important for organizing this complex. We also hypothesized that there may be molecular interactions between the proteins in this complex that were important for organizing it. And so to address that, we asked what happens if we start removing members of this little complex and if that affects others. So we got rid of the sortases and asked what happened to that localized general secretion machinery. And so here what you can see is that if we get rid of, for instance, SrtA, instead of seeing single foci of SrtA, again by electron micrographs, we start to see SrtA distributed around the cell. And we can quantify that down below here. So again, in wild type cells, most of them have single secretion foci. If we start getting rid of these sortases, this becomes more diffuse. So we think that molecular interactions are also driving the coordination of this complex. And finally, we started to wonder if these proteins and these highly positively charged cytoplasmic tails might be held in place by anionic lipid domains. And so right around this time, a number of groups were demonstrating that in gram-positive bacteria, there existed these anionic microdomains that coordinated secretion of proteins in other species. And indeed, in E. faecalis, we can also detect localized membrane microdomains. And here we're staining with a dye called non-acridine orange, which intercalates with anionic lipids. And we can see that they're in discrete domains as well. And so at this time, then we hypothesized that these anionic domains would also be involved in coordinating this process. Okay. So that's vignette 1, where we asked this question: why do these virulence factors appear in a non-random manner on the surface? And so our studies that I just showed you start to give us a glimpse of what might contribute to this ultimate pattern. And that is that at least the enzymes that are involved in the initial stages of producing these factors are all localized themselves. So it's a non-random process. Did we fully answer the question in the study? Absolutely not. That has been ongoing for a long time, largely driven by someone that's in the audience, Pei Chu, who's done a lot to figure out what really is driving this, but I will save that for another talk and focus on this nonlinear progression of little vignettes today. So we now understand that there is this localized domain that's mediated by anionic lipids and these positively charged tails and proteins. And so we asked the question then, well, okay, if we know something about what coordinates or governs the localization of this complex, can we use that information to disrupt it? And specifically, can we use cationic antimicrobials to disrupt these anionic lipid domains? And so to address that, we first started looking at antimicrobial peptides that these bacteria would see during a normal infection. We looked at human beta defensin 2 and 3. I'm just showing you the data for 2 here. We incubate the cells with sub-inhibitory concentrations of this defensin for very short amounts of time. And what we can see is that they target the bacteria in very discrete patterns. That is hard to tell in these images, but that seem to localize either at the septum or what will be the next septum in a dividing cell. You can much more clearly see that here in these electron micrographs. Again, here we're labeling with antibodies against HBD2 after this exposure. And you can see this massive accumulation of these defensins at single sites on the cell surface. So they are specifically localizing, targeting at discrete foci, as we predicted. So we then wondered if this defensin exposure could then disrupt this little complex. That was our initial question. So we first looked at these anionic lipid domains by NAO staining. So again, we see them sort of concentrated at domains at the septum in the absence of defensin treatment. Again, at the sub-inhibitory exposure level, we now see that these lipid domains, or what this dye is staining, becomes much more diffuse. And we can quantify that as well. So it seems to be disturbing these tight anionic lipid domains that we were measuring. I'm going to take a tiny segue and tell you about a protein called multiple peptide resistance factor. And the reason we started looking at this is known to be involved in resistance to antimicrobial peptides. So we're looking at how HBD2 disrupts these processes when we're wondering about other genetic factors that might help protect the cell from this focal targeting. And so what MprF is, and I'm going to come back to this in the second part of the talk, so this is important here. It's this membrane-anchored enzyme. This was first characterized in Staph aureus that has 2 functions. It adds cationic and amino acids to this very anionic lipid, phosphatidylglycerol. So it adds these cationic residues, and then it flips that to the outer leaflet of the membrane. And now we have made these anionic lipids cationic, and this serves to then repel electrostatically incoming cationic peptides. And so this is very involved in resistance to antimicrobial peptides in staph and other bacteria, including Enterococcus faecalis. And so we wondered if this had a role in that focal targeting of the defensin to the cell. And indeed it does. So here we're looking at the same exposures of HBD2, fluorescently labeled HBD2 with wild type cells. And so, or this MprF2 mutant cell. And these are matched exposures. And so we had to kind of turn down the fluorescence in wild type so that the MprF2 didn't blow out. And so what you can really clearly see is when we take away this cationic shield that's mediated by MprF2 in E. faecalis, now these peptides can target the cell even more strongly in this focal manner. And we can quantify that as well and complement that. So, so really this protein seems to be involved in the focal targeting by cationic antimicrobials. So the defensins target and redistribute anionic lipids. We also then asked if the same thing would happen to those focally localized proteins like the sortases and SrtC. And so again, we're now looking at the localization of these proteins here on the top. So this is SrtA on the left, SrtC on the right. And these little peaks are how we quantify the localized fluorescence. So each of those peaks is one side of the septum of this diplococcal enterococcal cell. So this is what we see in the absence of antimicrobial peptide. When we bring in the peptide, we now can track where the peptide localizes. So now this looks like it's targeting the peptide. And so that in the protein localization becomes much more diffuse. And so this tells us that that treatment with the antimicrobial peptide, the defensin, disrupts not only lipid domains, but also then the localization of these proteins that we think are coordinated in those anionic lipid domains. And so this was really interesting to us because around the same time that we published this first paper describing this in Enterococcus, another group in the United States published a similar paper in Enterococcus again, but now looking at daptomycin, which is also a cationic antimicrobial of a different structure, of course. But nonetheless, what they showed in that study was really interesting. So if they treated Enterococcus that is sensitive to daptomycin with these low levels of drug that will not kill the cells, they see this sort of spotty targeting. But at higher concentrations that are lethal to the cells, at very short exposures, they see a very different localization pattern. And they see that the daptomycin is really localizing or enriched at the septum of the cells. Again, that's where we see the antimicrobial peptides targeting. But in a daptomycin resistant strain, they don't see that targeting. They see that it remains diffuse. And so the argument in this paper was that one mechanism of daptomycin resistance is to prevent that focal targeting at the sensitive site in the cell, which is at the septum. Okay, so these looked similar, what we were seeing with cationic antimicrobial peptides and daptomycin. And so in the study, one of the things they did, so these daptomycin resistant strains are matched isolates from a single person that was infected with Enterococcus faecalis, underwent daptomycin treatment. And so this is a strain isolated, a sensitive strain isolated before treatment, and a resistant strain isolated afterwards. And when they studied this strain and did whole genome sequencing, they found a sort of constellation of mutations that conferred daptomycin resistance. And so no single one of these mutations conferred daptomycin resistance, but collectively they did. And so these were involved in a couple different functions in the cell. One is involved in sensing that daptomycin, that membrane damage, and then the subsequent transcriptional response, and that's shown in yellow or orange here. And then the other genomic changes associated with daptomycin resistant were associated with membrane remodeling, so changing the composition of the lipid membrane itself. So just keep this in mind for a second. I'm going to come back to this. So in vignette number 2, we then asked, can we target and disrupt the virulence factor assembly at these specific domains? The answer was yes, we can. That was exciting to see, and we can see these cationic antimicrobial peptides target specific spots, and so does daptomycin. And so we then wanted to ask a slightly different question, which was, can we use the what we're learning about how the cell reorganizes in these resistant strains? Can we try to understand that to better understand how these antimicrobials are specifically targeting the cells, and maybe help understand the underlying complex that it's targeting, this virulence factor assembly site, because that's ultimately, we're still really curious about understanding that fundamental process. Okay. And so here I'm getting into all new work. I haven't presented this publicly before. In the audience is the person who's done all of the work. His name is Zeus. So hopefully I'll do it justice, because this is a really cool part of the story. And so I've been mentioning daptomycin. So what is daptomycin? I told you it's a last line treatment for vancomycin-resistant enterococci. This is a lipopeptide antibiotic. It is cationic when it's complexed with calcium, and that complex is required for its activity. It's membrane targeting. The specific mechanism of action by which it kills cells is a little bit still unclear, but it certainly has an effect of extracting lipids in the membrane that it's targeting. So this is daptomycin. I already introduced to you MprF and how this works to mediate resistance to antimicrobial peptides, or cationic antimicrobials, I should say. And the reason I bring this up now is because, at least in Staphylococcus aureus, this is also associated with resistance to daptomycin. So DAP-resistant Staph aureus strains often have gain-of-function mutations in MprF. But this is not true in Enterococcus faecalis. So in human isolates of DAP-resistant strains, I don't think there's any reports of MprF mutants. And so this was curious to us, and specifically because Staph aureus has a single MprF gene, but E. faecalis has two paralogs of MprF, 1 and 2. And so as we were sort of thinking about this, so I come back to the mutations in E. faecalis that are associated in human infections with daptomycin resistance. I already mentioned these to you. But as we were thinking about MprF in sort of this broader picture of trying to understand how these peptides, these cationic antimicrobials are targeting the cell, we wanted to really understand what was going on and think about this a little bit more deeply. So we were thinking about MprF. So again, I told you MprF is involved in antimicrobial peptide resistance and DAP resistance in Staph aureus. E. faecalis, it's not, but we had a hint that it might be somehow involved in DAP resistance or DAP sensitivity in Enterococcus faecalis because an MprF mutant has a slight reduction in minimal inhibitory concentration for daptomycin. And so we started to wonder, maybe it was those two copies of MprF and E. faecalis that enable this pathway to not be targeted in the development of resistance because you'd have to mutate two genes in both MprF genes to get this gain of function. This was the hypothesis. And so stated another way, the question was, could this E. faecalis, these dual gene, dual encoded MprF activity, be masking other daptomycin resistance associated mutations? So maybe, E. faecalis has this cloaking mechanism mediated by MprF. Once we take this away, what's underneath? Can we learn more about daptomycin resistance, daptomycin targeting, antimicrobial peptide targeting once we take that cloak away? And so our hypothesis then was that there would be novel DAP-resistant mutations acquired when MprF was absent. And there's a corollary to that hypothesis, which was that DAP resistance should evolve faster in the absence of MprF. The cloak is gone. Now the cell has to respond more quickly to generate DAP resistance. Okay. So how did we address that? We performed in vitro evolution in the presence of increasing concentrations of daptomycin. So we start at sub-MIC levels, and over time, shown here is about 25 days for the slowest evolving strains, we gradually increase the concentration of daptomycin. And so what you're looking at here is sort of a color coding. Orange is low-level resistance, green is sort of an intermediate, and this blue is high-level resistance. And we've evolved them all the way up to this very, very high-level resistance to daptomycin. In black is wild type. In red is MprF1, that's one of the paralogs. You can see they look relatively similar. They evolve resistance, high-level resistance to daptomycin relatively quickly in the laboratory. And this really reflects what happens in human patients. And so while this is a last-line treatment for VRE infections, you can see that the window of time for effective treatment is often relatively short before resistance arises. If we look at an MprF2 mutant, we see that it's actually a little slower to reach that high-level resistance point. And then this MprF double mutant strain, I'll call this MprF null mutant, this evolves resistance to daptomycin much more slowly than the other strains put together. And so what this tells you immediately is that that corollary prediction that we had was wrong. This mutant is not evolving resistance more rapidly. It's actually slowing things down. But when we went in and did whole genome sequencing of different isolates along the way, our initial hypothesis did bear out. We started to see new, previously unappreciated mutations associated with DAP resistance in MprF mutant background. So important to point out is that we also did see mutations that are observed during human infection, evolution of DAP resistance in humans. These genes involved in lipid membrane remodeling, as well as sensing daptomycin. So we could also detect those in a wild type background. But what was really interesting to us is shown here. This first gene, this low-level resistance that we only observed in that MprF null mutant, and it was one of the earliest mutations we would see, and it's in this gene called FtsH. So this was what we were looking for, something new that we wouldn't have seen otherwise. So what is FtsH doing? And how can we figure out why this strain is evolving more slowly? What can we learn about DAP resistance? So to back up, what is FtsH? So this is a conserved membrane-bound hexameric protease. It's an ATP-dependent zinc metalloprotease for the protease aficionados out there. It is highly conserved in bacteria. And in many bacteria, it's actually essential. You cannot mutate it, bacteria will die. It is proteolytically active on unfolded membrane proteins, as well as SSRA-tagged cytoplasmic proteins. And while this is well studied in some model bacterial organisms, there's been no studies on this in Enterococcus faecalis. So we decided to try to understand why this mutation was popping up and its association with DAP resistance. So here is a cartoon of the different conserved domains of FtsH. This is the faecalis protein that looks very similar in other organisms. You can see there's transmembrane domains at the N-terminus. There's this ATPase domain, and then the protease domain. And so these little X's on top show where the different mutations that we detected in these resistant strains were arising. So you can see a lot of them are in the ATP binding domain, so this would definitely affect activity of the protein, a couple in the protease domain, and then we had this one at the N-terminus, which was actually a point mutation leading to a stop codon. So this was a total loss of function truncated protein. So we decided to try to work with this particular mutation and reconstitute this in the parental wild type strain that we like to work with. So to get rid of all of those other secondary mutations and really look at specifically the contribution of FtsH to daptomycin resistance. So we wanted to reconstitute this mutation. And so this is this version, this G37X truncate. And so when we tried to put this mutation back onto the chromosome in wild type bacteria, we found that we could not do that. And so that was consistent with a lot of literature saying that FtsH is essential. We couldn't truncate it, we couldn't mutate it in a wild type background. However, we could reconstitute it in this MprF null mutant background, which is where it originally popped up. But we could only do that when we kept daptomycin pressure on these bacteria. So we were able to do it, but we had to use some some tricks to get it to stick. But because we wanted to have this look at this mutant in a wild type background, which would be sort of the more natural way to study this, we took a second approach, and that was to express this truncate on a plasmid. Introduce that both into the wild type or the MprF mutant strain. And so when we did that, what we saw, so first in green is the MprF null strain, in this sort of the open green bar shapes are the MprF null strain expressing this FtsH truncate. You can see that this affects growth a little bit, but when we introduce that same truncate into the wild type cells, this has a much more severe effect on growth, but it seems to be a variable effect. You can see this with these large error bars. So it says that growth is sort of a mixed bag in the cells of these strain. And so we were a little bit interested in that. So when we plated the cells, they looked like something like this. So in this, again, wild type FtsH truncate expressing strain, we had some cells that grew into large colonies, some that were these small colony variants. And it turns out the reason for that is because in the colonies that grew larger, they had kicked out this truncate. And so the cells do not like to express that FtsH truncate. And so the ones that are growing very slowly, that maintain that truncate, there's some sort of burden or they're compromised in some way for expressing that truncate. And so this tells us that a truncated non-functional FtsH has a dominant negative effect on growth. And so what we propose is something like this is happening in this cartoon. There's a wild type version of FtsH on the chromosome, but then when we express on a plasmid this truncate, this somehow incorporates with this native complex to interfere with its function. So again, this tells us FtsH is essential for E. faecalis growth. And so one of the things we were just curious about is, well, what's affecting, or why is it contributing to this growth effect? Why is FtsH essential? And so we did a little bit of work to try to tease that out. In different organisms, FtsH seems to be involved in the degradation of a variety of different essential proteins. And so it's essential because it's important for protein control of other essential proteins. And so we wanted to understand this a little bit better in E. faecalis. And we took a for this, we took a transcriptomic approach, which might not be the most obvious thing to do at first, but this is what we did. And what we found was a couple of things. We saw sort of a net downregulation of genes involved in oxidative phosphorylation, a net downregulation of genes involved in amino acid biosynthesis and folate biosynthesis, and an increased expression in genes involved in glycolysis. And so what we think is that there's just this big metabolic reprogramming that's happening in this strain that gives rise to this growth defect that we were seeing. That's just an interesting aside. What we really wanted to know was, how is this FtsH mutation affecting daptomycin resistance? And is it specifically sufficient to affect DAP resistance acquisition? And so here we're doing that experiment. So we've now that we've characterized this truncate, we've reintroduced that again into this MprF null mutant background. And what we can see is that when we introduce that truncated FtsH, so we're now disabling to some degree FtsH in this strain, because there's still a wild type version on the chromosome. So, but what we see is that disruption of FtsH increases the rate of daptomycin resistance acquisition. So this was, as we would have predicted, because this was an early mutation that we found in the initial evolution, this is sufficient to increase the rate of evolution. And the reason for that, although we don't know why this is the case, is that FtsH mutation somehow affects the mutation rate in this strain. And so we can see, compared to the parental control strain, we see about a 2-log increased frequency in its mutation rate. So this starts to explain why this truncate or this mutation would be important for evolving DAP resistance. So I told you that FtsH is a conserved protease. And so if we want to know what the proteolytic targets of this protease are, to try to understand how it's contributing to daptomycin resistance evolution. So to do that, we hypothesized that if we looked at the proteins that are accumulating in the cells of a wild type strain versus an FtsH mutant strain, we should see FtsH targets accumulating in the mutant strain because they're not being proteolytically destroyed like normal. And so when we did that proteomic analysis, we did in the bottom see a number of proteins that were starting to accumulate when FtsH is absent. So these suggested these were bona fide targets of FtsH. We also saw a lot of proteins that disappeared, and this we weren't expecting at all. But nonetheless, this is what we see. We see some proteins that are less prevalent in the FtsH mutant strain. So we wanted to try to understand a couple of things about these proteins. Were any of these involved in, for instance, that growth defect that we were looking at? And so what we did here was we went to a more genetic approach. We disrupted the genes. So we now went to a wild type strain, disrupted each of these single genes that for proteins that were depleted, or we overexpressed genes for proteins that were accumulating, basically to phenocopy what we were observing in this proteomic analysis. And the first thing we asked was, do these genetic either disruption or overexpression affect the growth of E. faecalis? And indeed, we could find a handful did affect growth. And so again, these contribute, we think, then to that growth defect that we see when FtsH is inactivated. But again, that was an interesting sidelight, but our real question was, do these affect daptomycin resistant evolution? And so what we did here was that we started with these disrupted genes and we performed in vitro evolution. So one by one, we disrupted the genes and looked at how quickly they could evolve daptomycin resistance. And so here's just a handful of those mutations. What we see is that the bulk of them have no effect on daptomycin resistance evolution. A couple of them have small positive effects on the rate of evolution. And you can see that here, this the blue and the green strains on top. So if we disrupt those, we get slightly faster evolution. But really excitingly, what we found was that one of these genes, HrCA, had a negative influence on evolution. So if we disrupt HrCA, we slow down evolution. So this is where things, to me, start to get really exciting, because this is what we're looking for. We're looking for what is responsible for this slow to slowing evolution to daptomycin resistance. So what is HrCA? I think HrCA. Why is this so interesting? So this, again, is a pretty conserved protein. Is actually a transcriptional regulator, and it's a negative regulator of a variety of chaperone operons that are involved in stress responses, heat shock responses in particular. And so it negatively regulates these operons. And so when this is mutated, the prediction would be that this repression should be relieved, and you should see an increased expression of these different chaperone proteins. And so there's been a couple of papers recently that have described similar things, that there's either an induction of chaperone genes and or an association with their contribution to rates of evolution to antimicrobial resistance. And so we're very excited now to look at this a little bit more closely. And this is particularly exciting, again, because if we can really figure out how HrCA is slowing down antimicrobial resistance acquisition, this is a potential new therapeutic target. So, similar, at least in idea or in concept, to something like clavulanic acid that you can, treat with an antimicrobial and something at the same time that will prevent or slow evolution of resistance to that antimicrobial. So increasing the therapeutic window for infection. So this is where we're at with this. And so just coming back to the beginning of this last vignette, we asked if MprF activity, this dual gene encoded activity, could be masking other resistance-associated mutations. That seems to be indeed the case. We found that FtsH is essential in E. faecalis, and mutation of it accelerates the rate of DAP resistance acquisition. But we also found these other genes that can conversely slow acquisition. And so just to summarize this last little piece, so what we found then again is dysfunctional FtsH. We've learned some just fundamental things about FtsH in E. faecalis, how it contributes to normal healthy growth. I won't really summarize that, but what we found was that by studying this fundamental process, we were able to, in the context of DAP resistance, we were able to identify this new gene that could slow DAP resistance evolution. And so we sort of put this all together. We have a lot of questions. We want to understand these metabolic effects better. But for me, really the key question for this talk is this lower right square, is whether now this chaperone activity that I mentioned that HrCA seems to control, at least in other organisms, if that's responsible for this delayed DAP resistance evolution. And if so, then we can think about how to turn up chaperone activity potentially as a way to thwart resistance evolution. And so that brings me to the end of this little series of vignettes where we asked if we could try to use this basic studies of antimicrobial resistance to better understand interactions with these antimicrobials as well. And yes, that we could do this, this has really led us to this really exciting new place where we finally at this place where we can start to think about how we might interfere with this potentially targeting and killing by these antimicrobials. And so this really then is my take-home point, that this is a long series of research by many people, but it's basically a basic research path that is curiosity driven at the beginning, that enabled us to finally get to a place where we have potentially translational conclusions that might be important for treating these infections, which of course is the ultimate goal, as I told you in the beginning. And so with that, I'll stop. I showed a number of published papers, and those people are long gone from the lab, so they're not in this picture anymore, but their names were on all of the slides. And really, I want to highlight Zeus Nair, who is a soon-to-be graduated PhD student in the lab, who did all of the work in the second half with the DAP resistance and FTS. So with that, I will stop and be happy to take any questions.
Thank you, Kimberly. Very nice talk. Beautiful work. Very enjoyed. So, in the early, in part of your talk, you mentioned Sortase-A. we as a chemist, we are interested in sortase as a tool, for protein modification ligation. That looks like it's a very important enzyme for these gram-positive bacteria all have this ligase, right, sortase, right?
Yeah.
So it looks like it's
exactly.
very essential, right, for the cell wall attached proteins.
Yeah, it's not essential for bacterial growth, but it's essential to put those proteins on. And many of those proteins are really important for interacting with host cells or forming biofilms. So the sortase attached proteins are important, but there's only one example, one species I know of where the sortase enzymes themselves are essential, suggesting that they add something to the wall. It's integral for the bacteria to grow. But in most gram-positives, it's really just involved in virulence factor production. And so this makes it a interesting anti-virulence target, right, rather than a antimicrobial, killing target.
Mm-hmm. Great. So I think there's a question from did you see it? I think from Oliver.
Yep. Can I talk?
Sure.
Yeah. That was a really great talk, Kimberly, and lots of really interesting, a lot of lines. I find it interesting, this that the delta HCRA seems to evolve more slowly. So what I'm wondering, I mean, if I would expect that such a strain, it'll overexpress the chaperones. Of course, there should be a trade-off maybe with growth rate. So then if that is true, if the growth rate was a bit slower, how can you, maybe the generation time is longer, how does it relate to evolution rate, so to speak? Can you, yeah.
Yeah, so this is pretty new data. So these are experiments that are ongoing. So we don't see any transcriptional induction of those chaperones. So that doesn't make sense to us, at least by RNA sequencing. So we need to go back and do more careful transcriptional analysis, because that would be the prediction. So that's point one. We also, on deck, I think actually ongoing right now, is the evolution experiments in those mutant backgrounds to address that question exactly. Because we don't know, HCRA could also have other targets in E. faecalis that might be contributing to this. So these are actively under investigation.
Great. And then I have another quick one. So it's very interesting. So you think in the end, this localization of these, with the sortase and the polylysine tails, it's due to the distribution of anionic lipid domains in the in the membrane. So I'm wondering, what do you? So basically, of course, that will depend on heterogeneity in the membrane. And what do you think, or what do we know about how that heterogeneity comes about? Because then, of course, you're shifting the problem of the localization to the membrane, so to speak, and that needs to be somehow brought about.
Yeah. So these are really important questions that we're also investigating, and it's pretty complicated to look at. So the storyline I gave you was sort of the very naive way we were thinking about it as we went. We've also been doing a lot of work to try to understand membrane composition, what those microdomains might be. That dye we're using, it's supposedly stains cardiolipin. It's very non-specific, though. We start to deplete essential anionic lipids. We can, delete cardiolipin synthases. So we do those sorts of genetic experiments. But then, of course, there's all these other pleiotropic effects that we're seeing. So those are actually a little bit hard for us to tease out. The other problem with that hypothesis, that this is really coordinated by anionic lipids, is the vast majority of the membrane, something like 90% or so in this class of gram-positive bacteria, are anionic lipids. So then it's got to be a subset of some, a subset of lipids. And so we're looking at things like membrane vesicle production, which we think pinch off of the septum of the cell, to try to understand if there are microdomains of specific lipids that coordinate those processes. And that's actually a project Zeus has been working on as well. And we do see different lipid compositions associated with the sortase enzymes, for instance. And so that's how we're trying to understand that. But there's still a lot for us to understand here.
Thanks.
Any other questions?
Can I just ask a question?
Sure, yeah, go ahead.
Hi, very nice talk, thank you. Do you in the wild, I mean, during the patient populations, ever see instances where there's loss of these MprF genes in response to treatment?
Not that I'm aware of, not that's in the literature. So in like Staph aureus, we'll see these gain of function mutations in DAP resistant strains, but in E. faecalis they just don't show up. And again, we think this is because there's two copies essentially, and we've been trying to understand how the two copies contribute this, to the function of MprF and E. faecalis. But at least we postulate that that's why we're not seeing them in a patient, because we might, somehow like a mutation in one is not sufficient to confer resistance. We also don't see, some of these other mutations we're picking up in these weird genetic manipulated type experiments. We're not seeing those in people. And certainly some of these mutants that we're discovering, we wouldn't show up in people because they're not contributing to resistance. They're actually slowing resistance. And so I think that's an important point, is that, by doing sort of these convoluted bacterial genetic experiments, we can find things that you would never see pick up in a patient, that might be useful to know. So.
Right. Thank you.
Okay, any other questions?
Can I ask another question since no one else is asking?
Sure.
Yeah. So this sort of is related to Oliver's question. So about heterogeneity of these anionic domains. So you have these antimicrobials like defensins, which are largely positively charged. And then, you have these things responding by adding lysines and then flipping the lipids over. Do you think there's any mileage in wondering whether this modification happens in certain regions? And then you may also have peptides where you have a combination of positive and negative charges, which might find some therapeutic window. Does that make sense?
Yeah, so you're asking if these modified lipids are somehow localized in the cell, and if you can sort of bypass them and target other parts of the membrane?
Partly, but also your peptide will now also have negative charges, which will approach these. It's a balance, isn't it? An electrostatic repulsion. Yeah. But can the negative charge peptide approach the lysines, sort of smother them out? And then.
Yeah, that's interesting. I suppose that's possible. We've done a little bit of work comparing like naturally occurring anionic antimicrobial peptides to cationic ones, and they are not very active against the bacterial cell, but that's different than what you're asking specifically. I guess theoretically, if you could somehow make the first point of interaction anionic and then the second one cationic, that you could sort of do a dual step targeting. That is potentially interesting. But again, we don't have a good sense of if these modified lipids are localized in the cell or not. That's kind of hard to monitor. We can monitor the location of the MprF enzymes themselves, and those seem to occur at microdomains, but that doesn't say that's where the lipids, the flipped lipids are retained.
Right. Okay, thank you.
Thank you. Any more questions? I think Prof 10 has a question.
Can you hear me?
Yes.
Thank you. I have a lot of problem with my internet. Kimberly, thank you for a wonderful talk and excellent work on understanding the mechanism of bacterial infection. I have a very general question. It looks like cocktail probably will be the trend to overcome bacterial infection, because bacteria continuing to evolve. Is it possible to design a molecule hopefully not too expensive antimicrobial peptide that can incorporate some of the characteristic of cocktails and I mean antimicrobial cocktail. Now you use daptomycin as an example. So specifically, can you improve what aspect or what area, what part of structural motif would you advise synthetic chemists to improve to give out to overcome bacterial infection, specifically the topic you're mentioning too?
Yeah, well, I'm not in any way, shape, or form a chemist, so I don't presume to suggest what a synthetic chemist should do. But I think what I can comment on is what would be potentially interesting. Like if one could link up, and and there's plenty of people doing, as linking peptides to different molecules. But if for instance, if we take this mechanism that we're still unraveling that slows DAP resistance, if we can figure out what that is, and then figure out a way to turn chaperones up or whatever, affect that to a small molecule that can give the same effect. If we could somehow potentially link those two, because to my mind, the peptides will sort of target the membrane and probably get this molecule through the membrane and potentially deliver it to its cytoplasmic target. That's one way I've been thinking about that. So it's a dual therapy, but a conjugated dual therapy. But again, this is pretty preliminary, just sort of a thought exercise for us at this point.
Yeah. Well, thank you. Thank you very much.
So any more questions? If not, I want to thank Kimberly again for a very, very nice talk. It's kind of fascinating. This bacteria can evolve resistance even against membrane acting, peptides. And people used to think that it's difficult for bacteria to evolve resistance, against this membrane targeting peptide. But yet, they can do. It's very, really, very fascinating. And I think Kimberly's work has revealed and give us many interesting targets. And for us as a chemist, for me, to discover, drugs against these, new targets. So with that, I want to thank everybody for attending. And for your information, this seminar is recorded. So if any of you or your friends, are interested in watching it again, review it, you can ask Elaine from the Institute of Advanced Studies for the video link. Okay, with that, I want to thank you, everyone, and also especially thank Kimberly for a wonderful talk.
Thank you.
So you have anything to say?
Me?
Yeah, and I'll thank Elaine for organizing the whole thing.
No problem. Thank you, Prof. Kline, for accepting our invitation.
Mm-hmm.
Thank you.
Thank you. Yeah.
Okay, thank you, everyone. Bye-bye.
Bye-bye.
Thank you, Kimberly. Wonderful talk.